Hi everyone. This is a short video to cover the material that got chopped out of our recording from the session on Monday, August 31st. When I split everyone into groups, the recording turned off. We were chatting about this particular graph from your lecture outline before the recording stopped. So I wanted to take a moment and make sure I covered that material with you that we chatted about at the end of that class session. This particular graph is what I like to call the recipe for the fluids that are in the body. Or in other words, if I wanted to make these kinds of fluids in the body, what would I need to add to them? Now, remember from our lesson on Monday that there are kind of three main types of fluids that we're focusing on in the body. The first one is our intracellular fluid. And if you recall from that lesson, the specific name of intracellular fluid is cytosol. So my first type of fluid in the body that I might want to make or that I have in the body is called cytosol. That's an intracellular fluid. The second type of fluid in the body that we have is called interstitial fluid. And remember that interstitial fluid is found in between the cells that are in our tissue. It is an extracellular fluid, which is what this ECF means, extracellular fluid. The last kind of fluid that we focused on this graph is plasma. And remember that plasma is found outside of my cells, inside a blood vessel. It is also an extracellular fluid, meaning it's found on the outside of my cells. When I look at this graph, this is what we call a bar graph. So the size of the bars on this graph give us an idea if we have a lot of something or just a little of something. So as we work our way through this graph, we can see that there are different amounts of things inside the cell. So inside the cell, we are representing these types of things with this beige bar. Their amount is shown with the beige bar. Things that are found either in between cells in tissues or things that are found inside plasma, those are represented by the blue bar and the red bar. So as we start looking at this graph from the left side, the first thing on this graph is sodium. Sodium, notice that the bars are very tall for blue and red fluids, meaning I have a lot of sodium inside interstitial fluid, represented by this blue bar, and inside plasma, represented by this red bar. Notice that inside my cell, in cytosol, there's not very much sodium. Next, I go over to chloride, Cl-. Notice that the bar for intracellular fluid is still very small, not very much chloride inside the cell. But outside the cell, whether we're inside tissues or whether we're inside plasma, that level is a lot higher. So lots of extracellular sodium and chloride, not as much inside the cell. But notice when we move to potassium, notice how tall the bar is for intracellular fluid as it relates to potassium. Inside the cell, we have a lot of potassium. Not so much outside the cell, whether we are inside interstitial fluid or inside plasma, not very much potassium. So potassium is focused inside the cell. The last category on this graph is proteins. Notice that I have a whole lot of proteins inside my intracellular fluid. Now, granted, the concentration isn't as high as we see of some of these other ions, but it's still much higher, that concentration in cytosol, compared to the concentration in interstitial fluid. We discussed how we would also say that plasma has a high level of proteins as well, at least high compared to interstitial fluid. In fact, the main way you can tell the difference between plasma and interstitial fluid is that number of proteins that they have. Those are proteins, like your guided lesson explain, do things like blood clotting or do things like immune reactions. So we would say that our protein level is high in plasma as well. What we talked about to help us remember this information about where I find various things is the example of what we'd call a salty banana. As in, think about a banana that you sprinkled salt on the outside of. That's what your cells are like. They are a salty banana. The reason that this particular analogy works for us is because a salty banana has the stuff on the outside and the inside that your cells have. So when we think about salt, here's a little chemistry for us here. The chemical formula for salt is sodium chloride, or in the abbreviations we saw on the previous slide, Na plus and Cl minus. When we were talking about what we found a lot of on the outside of a cell, we had a lot of sodium and a lot of chloride in those extracellular fluids. So if I sprinkled salt on the outside of my banana, I'd be putting a lot of sodium and chloride out there. That banana itself is really high in potassium. Potassium is the ion that we see a lot in bananas. Potassium was K+. Bananas always have a lot of potassium in them. That is something that they're known for. And we have a lot of potassium inside our cells. The other thing that bananas have a lot of inside of them is glucose, or sugar. As you complete lesson number 2, you'll see that one of the other things that we have a lot of inside our cell is glucose. Our cells really like to grab glucose because that's what they're going to use to make energy. So bananas have potassium, bananas have glucose. We discussed during our class time that we also want to pretend like this is a super banana. So if bananas were absolutely perfect, the one other thing they would have a lot of inside of them are proteins. So as you're considering what I have a lot of inside or outside a cell, think about a super banana that happens to be salty. On the outside of this super banana, I've got a lot of sodium and chloride. On the inside of that banana, I've got a lot of potassium, glucose, and proteins. That wraps up the discussion that we had on Monday, August 31st. We will keep about lesson number 2 material on Thursday, September 3rd.